right, guys, this is chapter 7. We're going to begin the nervous system physiology now. So this is nerve cells and electrical signaling. So we're going to be talking about neurons and how they physically send an action potential to another neuron. So let's begin by looking at the anatomy of a neuron. So I'm going to go over this first part relatively quickly because this is review from anatomy. So we have our two neurons here that are forming a synapse. Remember, a synapse is a communication point between two cells. It doesn't have to be two neurons. Whenever a neuron communicates with another cell, it's a synapse. Synapses will always have a gap between the two cells so they don't physically touch, and that's called the synaptic cleft. The soma is the body of a neuron. We have an axon hillock, which is a bulge where the axon begins. After the axon hillock, I'm just going to add another section here, and this is called the initial segment. So the initial segment is where your action potentials will begin. We know that the dendrites are receiving input from other cells. So the dendrites are receiving information. The axon then sends the information away. So here's the direction of our signals, right, moving away. And at the end, the axons will split. And those are called terminal arborizations, if you remember that from anatomy. And then we have the axon terminals, which can communicate with one or many neurons. Sometimes axons can split and branch, and they're called collateral axons if there is a branch. Most of the time, dendrites are the highly branched processes coming off of a neuron, and axons, there's usually one very long process that goes to the effector organ. So these are just some nice pictures of dendritic branching. So you can see all the very highly branched network that are made with these dendrites. In terms of the localization of ion channels in neurons, so we'll be talking about this a little bit later, but dendrites in cell bodies will have most of the time these ligand or mechanical gated channels. So if we want to just add that note here, so in this region of the neuron, we'll have either ligand gated or mechanical gated ion channels. And then in the axon, we're going to have this region, we're going to have voltage gated ion channels. Okay, and that'll become important in a little bit. And the initial segment, which is right next to the axon hillock, as I mentioned before, has the greatest amount of action potential. So if I were to sort of just create a little axon hillock or initial segment here, initial segment, where your action potentials begin, this is where you have the greatest amount of voltage gated ion channels. Okay, so just that's the first part of the notes. For axonal transport, like we're looking at here, that just means that we're transporting substances that are made in the body of the neuron, or transporting towards the terminal axon terminals. Neurotransmitters are transported this way. So if we look at the neurotransmitter walking here, so neurotransmitters are created in our soma with the organelles that are found there. They are packaged up in the synaptic vesicles. So your neurotransmitters are inside here. And then they're going to be moved physically by a protein called kinesin. Okay, so this is our kinesin protein that literally walks. So these two parts of the protein take conformational changes that look like they're actually taking steps down the microtubule. And the microtubules are acting like the railroad track that the kinesins are going to move down. And so you can see that it takes a lot of energy to do this. The synaptic vesicles are going to be moved down to the axon terminal. The axon terminal is where these vesicles with the stored neurotransmitter will rest until they're needed. And so when an action potential occurs, there is exocytosis where the neurotransmitter is secreted from the axon terminal, and sometimes it's taken back up, but then we can also transport in retrograde. We can transport substances back towards the soma. Okay, so anterograde is moving sort of from the soma down to the axon terminal. Retrograde movement is from the axon towards the soma. All right. Let's take a look at some anatomical morphologies of different neurons. You've seen these before in anatomy class. So this is bipolar neurons. Bipolar neurons have a cell body that's situated in the center, and we have processes that come off either side. A pseudo-unipolar neuron basically has one uninterrupted axon. So you can see that both sides are considered axons, and the cell body lays off to the side. And our multipolar neuron is going to be highly branched in the dendrites on the soma, and one very long axon leading away from it. So those are just the structural classifications of your neurons. Neurons functionally have different functions to them. So this should be also a review, right? So sensory neurons are also called afferent neurons, and those neurons will bring sensations or action potentials into the central nervous system. So coming from the periphery of the body, the peripheral nervous system, right? So these will be your sensory receptors of the body, of your skin, bones, your muscles, joints, connective tissue, as well as the viscera, so your organs. Anything that comes into the central nervous system as a sense, right? So the afferent or sensory neurons have that function. They will connect into the central nervous system. There are a network of interneurons that help to create complex pathways so that the sensory information coming into the central nervous system can find the correct pathway. So interneurons will create complex pathways. And then if the central nervous system wants to send a message outgoing, right, to an effector organ, then the neurons that do that are called efferent neurons. These are also sometimes called motor neurons, right? And they will lead to some kind of effector organ. So this can be skeletal muscle or smooth muscle, the smooth muscle and glands. So anything that will have an effect. Okay, so the output runs through efferent neurons, whereas input runs through afferent neurons. All right. So I do also mention in this first part to review your glial cells. There is an area in the textbook where you can review your glial cells, and also review what's involved in the central nervous system and peripheral nervous system. All right, now let's talk about the establishment of resting membrane potential, and we are going to look at first equilibrium potentials of both potassium and sodium ions. Those are the two ions that are going to be critical in the conduction of graded potentials and also for action potentials. So if we look at the membrane potential to potassium, so let's start with potassium. So let's start with number 1 here, where we see there is an equal amount, right, 150 millimolars of potassium chloride and 150 millimolars of sodium chloride. So for simplicity's sake, we'll just say potassium and sodium, since chloride is the same on either side. So we're going to see some gated ion channels here in blue. And if there is no charge difference and no concentration, if there's no charge difference, the membrane potential is at 0 millivolts. All right. So what if we opened the potassium channels? Because potassium has a higher concentration inside the cell than outside the cell. Okay, we'll just put a small potassium here. There's a very large potassium concentration inside the cell. Then the potassium will want to run down its concentration gradient and move out of the cell. Now, as the potassium is moving out of the cell, because potassium has a positive charge, right, we're having positive charge leave the cell, and so we're going to leave the internal environment of the cell slightly negative. Let me just change colors here. Right? So as potassium leaves the cell gradually become negative. All right? So at some point, so the concentration gradient is going to move potassium out. Because there's no charge difference at first, there's no electrical force pulling potassium one way or the other. However, as the membrane, right, if we go to number 3, as potassium leaves the cell, the internal environment becomes more and more negative, and that's going to pull some of the potassium to move back into the cell. So we can see that the concentration gradient is moving potassium out, but the growing negative charge inside is going to be pulling the potassium back in. So at what point do we have the outward efflux of potassium going out? At what point, what electrical charge, right, or what membrane potential would it equal potassium flowing back in? So if we look at number 4, right, eventually the same amount of potassium ions will enter as well as leave the cell. Right, so we're going to have an equal and opposite action of the concentration gradient and the electrical gradient of potassium ions. And so what we say is when this point has been reached, when potassium is equally flowing out and into the cell, that is the equilibrium potential for that ion. So the equilibrium potential of potassium happens to be around negative 94 millivolts. So the cell has to be quite negative for the potassiums, or at negative 94 millivolts, right, there's an equal flow of potassium out as well as back into the cell. All right, let's look at sodium. At rest, okay, so if we're establishing a resting potential, so here, so actually let me back up and just talk about the equilibrium potential for sodium. It turns out that the equilibrium potential for sodium wants to move into the cell through both the concentration gradient and the electrical gradient. So sodium, because it has a larger concentration outside the cell than inside the cell, the diffusion gradient, the chemical gradient, is going to pull sodium into the cell. And if the cell has a negative resting potential, as you see here, then the electrical gradient is also going to want to pull sodium inside the cell. So instead of potassium, where we have opposite opposing forces, where their concentration gradient wants to move potassium out and the electrical gradient wants to pull potassium back in, sodium, actually both forces work in the same direction. So at rest, the cell is at negative 70 millivolts. If we open just the sodium channels, right, sodium is going to enter the cell due to both its electro and chemical gradient. As more positive sodiums come in, right, then the interior of the cell will start to become more positive. And so sodium will also want to leave because the like charges are going to repel each other. And so it turns out that the equilibrium potential for sodium is going to be positive 60 millivolts. So we see that down here on our little chart here, the equilibrium potential for potassium is negative 90 millivolts. And you know what that means now? You know that when potassium enters and leaves the cell at the same rate, that happens when the cell's membrane potential is negative 90, so it's a very negative interior. When the same happens for sodium, if we just look at sodium, the equilibrium potential for sodium is going to be a positive 60 millivolts. Okay, so at a positive 60 millivolts, this is when sodium will flood in and also leave the cell equally. So that is the point we wanted to do here. So sodium moves down its concentration gradient until the electrical gradient or positive charges oppose further movement of the sodium. All right. So All right, so now if we allow both potassium and sodium ions channels to open, the cell's membrane potential will lie exactly between the equilibrium potential for sodium and the equilibrium potential for potassium, right? So I put a little arrow approximately where that would be. So somewhere in the middle between the equilibrium potential for sodium and that for potassium, in the middle would be if they were both open, right? If both potassium channels and sodium channels were open. However, the cell's resting potential is slightly more negative, right? So we can see here the resting potential is actually not between the two, but it's much more negative than that. And the question is why? Okay, so this resting potential, establishing our resting potential at about negative 70 millivolts, what contributes to this? So there's a couple things here. So first of all, we know that the sodium-potassium pump, otherwise known as sodium-potassium ATPase, right? Because this directly hydrolyzes ATP. It breaks down ATP to ADP and P to work. So it is a primary active transporter. This is going to help establish the chemical concentration gradient. So it's going to pump, we can see the pump here, right? It's going to use ATP and it's going to pump 3 sodiums out of the cell, right? So this is the intracellular fluid and the extracellular fluid. So this side is inside the cell, this side is outside the cell. So it's going to be pumping 3 sodium, 3 positive charges out, and it pumps in 2 positive charges. So because it's pumping out 3 positive charges and it's pumping in only 2 positive charges, then we are leaving a little bit of a negative charge, right? So the ATPase, the sodium potassium pump, does let a little bit of a negative charge remain inside the cell. But also, we want to look at the leak channels. So the potassium and the sodium leak channels are going to allow these ions to diffuse down their concentration gradient. Now, the cell actually has more potassium leak channels than they have sodium leak channels. So we can see here, we have 2 potassium leak channels being shown. We only have 1 sodium leak channel being shown. So we're allowing more positive charges to move out, right? Potassium wants to move out of the cell, so it leaks outwards, which means that we're allowing more positive charges to leak out, which again leaves us with a negative interior. And there are fewer sodium channels coming in, so fewer positive charges coming in, which means that the resting membrane potential will be negative. Okay, so between the action of the sodium-potassium pump, which has an unequal pumping of charges, right? It pumps more positive charges out, leaving the inside negative, and the fact that there are more potassium leak channels, potassium wanting to leave the cell, positive charges wanting to leave the cell, leaving the interior of the cell negative, fewer sodium channels leaking sodium in. Therefore, between these two, we have the resting potential. So nothing's going on when a cell is doing resting, right? There's no action potential, but we've created a membrane potential at negative 70 millivolts. So there is a potassium and sodium concentration difference, and there's also a charge difference with the internal environment of the cell being negative 70 versus the outside of the cell. All right, so let's see if we can answer this question. Why does the resting membrane potential tend to be closer to the equilibrium potential of potassium rather than that of sodium? Okay, so remember that if we open only potassium channels, potassium's equilibrium potential is going to be at negative 94 millivolts. And if we open only sodium channels, the equilibrium potential for sodium is going to be a positive 60 millivolts, right? But true resting potential is actually at negative 70 millivolts. It's way closer to that of potassium, right? It resembles more, we're going towards equilibrium potential potassium. And why is this? It's because of letter C. The membrane is more permeable to potassium, So it's kind of like thinking if all our potassium channels were open, except they're not all open, but it is leakier to potassium. So just overall, more potassium is going to move down its concentration gradient until it wants to reach equilibrium potential. So that's why the resting potential is going to be closer to the equilibrium potential of potassium. All right, so that was part 1.